We're going to start our semester off by beginning to understand what is biology. And to appreciate biology, we got to know what life is and what it's all about. So today I'm going to go over kind of three big characteristics or three big topics for our understanding. They are what are the characteristics of life? We're going to look at viruses and the role they play, and we're going to go over some basics about the microscope. Now you may have been given one or two of the following textbooks, but either one you should be able to turn to chapter 1 to read through it to kind of revisit these concepts and get a stronger understanding and help you fill out your study guide as well. So the first part I want to talk to you about is the scientific method. Now what I've done to help you with your study guide is I provided you a page number up in the upper right hand corner if the material I'm going over can be written down in your study guide. So you can see here it says SG Study Guide page 2. So everything we're going to cover you should be able to write down on page 2 of your study guide. There are certain steps that we need to take for the scientific method. The first thing we generally do in science is we make observations. We're looking at what's going on around us. We're asking ourselves questions, why did that happen? And then we try to explain it or figure out what event just occurred. So when we make an observation, that tends to lead to a question, and we can form the question that we want to answer. And when we get that question, it allows us to create a hypothesis. And a hypothesis is a testable statement. They're generally written down as a if-then statement, and I have some examples that we're going to look at. After we get done with our creating our hypothesis, we are going to develop an experiment. And in our experiment, we have different types of variables and groups that we use to figure out if our hypothesis was correct or not. So let's look at our experimental design. We always need a control group. The control group is the group that we're not going to do anything to. It provides the standard for comparison. It's the baseline, so that when I do the experiment, I know if what I change actually worked because I have this group over here I can compare it to. The experimental group is the group that we are going to mess with. We're going to change one thing to them, and we're going to let our experiment run, and we're going to see what happens. And that one thing we tend to change is called the independent variable. It's the thing we're manipulating. It's the thing we're going to do to the experimental group. Now, sometimes in an experiment, you have to, quote unquote, mess with the control group because you don't want the control group to know they're not actually getting the experiment ran on them because it would kind of mess up your results or your data. So for example, say I'm testing a new form of medicine and I want to know if my medicine works. Well, I divide my population into two groups, control and experimental, and I would give the medicine to the experimental group. But the control group needs to be given something, and that's what we call a placebo. The placebo is like a fake independent variable. The group will think they're getting it, but they're not, and they won't know they're not getting it because I don't want them to know they're getting it. It's called a blind study. So they get this placebo, and it could be actually something like a sugar pill. So the control groups think they're getting the medicine, the experimental group thinks they're getting the medicine, and they really are, and then I can see, well, does my medicine work? And using the placebo gives the same thought process in the control group, so my data will be much better and more valid. The dependent variable is pretty much the outcome. I messed with the independent variable. I want to see what happens, and what happens is the dependent variable. This is what I'm looking for when I run my experiment, and this is what I'm going to measure for or observe for during my work. Once I have ran my experiment, I need to document my data. So I got to see what happened with that dependent variable. Maybe I'll put it in a data table, maybe it's something I can graph, but there's two ways to look at collecting data. One of them is to just make observations using your senses, which would be what we call qualitative data, and the other one is where we can actually measure the value of the data, and that's quantitative. Once I get my data, I need to look at it and kind of reflect upon what happened and decide, was I right? Was my hypothesis correct? Yes, no. And you know, the nice thing about science is it's okay to be wrong, because even if you're wrong, guess what? You still got data. Now you know what not to do. So that can be part of your reflective process when you sit up there, you sit there and you're looking at your data and like, man, this is really wrong. Wrong, bad, isn't anything to worry about. It's still data. You still got results. You still have something that you can think about and reflect upon in your conclusion. So here's those scenarios I was telling you about earlier. Let's say I was making an observation that, hey, my flashlight doesn't work. I wonder why. Well, one idea is the batteries are dead. So I could create a hypothesis that says, if I replace the batteries, then my flashlight should work. So what I do, pop in some new batteries, I look and see, nope, it didn't happen. Was I wrong? Yes. Is it okay to be wrong? Yes, because guess what? I got data. I know now it wasn't the batteries. So this can let me form another idea that maybe the light bulb's burned out. So my next hypothesis could be, if I replace the light bulb, then the flashlight should work. So if I run through my experiment and I swap out light bulbs and it works, hey, I got it right. It just took me 2 tries, and that's okay. So when we do use the scientific method, you have to make good observations. You have to pay attention to the process and what you're doing, because you don't want to miss any details. You're watching, you're observing, you're getting your measurements, and you're being a good scientist in getting that data collected. One thing we try to avoid is inferences, because that's too much along the line of making assumptions, and we really don't want to make assumptions. We want to stick to the facts and the things that we can see and measure when we come to our conclusions and writing up our summaries for labs. This leads me to theory, because a lot of times in science you come in with the idea that a theory is a hunch. I have a theory about something, and we don't want to use the word theory like it's a hunch. It's not a hunch. A theory is a proven, time-tested, strongly supported experimental process. It's a hypothesis that has been tested and retested and tested and retested, and it always has good, consistent, predictable outcomes. There's very little variant in there, and so it's something that we fully believe in, and it's strongly supported, and we consider it to be true. So that's a theory. It's not a hunch. Laws, on the other hand, are absolute concretes. There's no give in a law. It's a fact. If I pick up my pencil and I hold it over the floor and I let go, it's gonna fall to the floor. That's a fact. It's called gravity. So there's a minute difference between theories and laws. Theories we can't, will eventually hopefully become laws. They're just not quite over there yet to where it's a concrete fact, but they are truly strongly supported in science. They're not hunches at all. So now we're going to start moving into this whole thing about biology. And we're looking at page 3 now on your study guide. So biology, believe it, is the study of life. So what is that about? Well, we don't just study like what life around us right now. We study the history of life. We look at life in the distant past. We look at life on the microscopic scale. We're exploring the solar system to see if there's evidence of life. So we're looking for life all around us, whether it's past, present, shape, size, form, place. We're looking for it. And it's a pretty complex thing when you start looking at life and trying to decide, well, what is living or not living, because there's different properties and characteristics that we need to examine. What we do know is that there's 3 big features, 3 key features that all living things share, regardless if they're microscopic, regardless if they lived 100 million years ago or 3 billion years ago. They share these 3 things. And one is, if you're a living organism, you need energy, which means you need nutrients so that you can get the energy from your nutrients. So you got to have a source of energy. We also know that you need to be able to maintain something called homeostasis. Homeostasis is your happy place. Homeo means same, stasis means state. So you're maintaining your same state. And that's a small window depending on the organism, because if you go one way too far or one way the other way, you'll die. Okay, so you're trying to maintain this constant state within your organism self, like within your body, and we'll talk about that in more detail. And then all living things have this wonderful molecule called DNA, and DNA is what lets you be you. It has the recipe, it has the directions, it has all of the information so that you can be you. That's it. That's a blueprint for life. And we actually devote all of second term to understanding and appreciating DNA. That's how important it is. So now we're going to look at characteristics of life starting on page 4. And one thing about organisms is you got to be organized. We're not just some random collection of parts. There's a pattern to our systems in our body and right down to the cells. And so we need to look at that and understand that how are we organized. One other thing is that all living things are made up of cells. That's like the clincher. If you want to decide if something's living or not, ask yourself the question, is it made out of cells? It can be just a single-celled organism, or it can be something that's multicellular like you, which has trillions of cells. But if it has cells, then it's considered a living organism. It's a living thing. So when we look at organization, you can see that, hey, I'm not some random assemblage of parts. I have structure. I have function based upon the structure, and that goes right down to the building blocks of all living things, and that's chemistry. We're made out of atoms first and foremost. You look at the periodic table, you can see the chart of elements. We're made out of that. Carbon, hydrogen, oxygen, nitrogen. Those are the 4 main elements that make up your whole body. It's like 96% you. What I can do with those atoms is that I can build upon them. I can grow them and make them bigger by forming molecules. And molecules, again, it's a little bit more complex than a single atom. But when I start getting molecules, I can start creating a little bit more diversity and function in what those molecules can do. And we have actually 4 basic type of molecules that make up your body: carbohydrates, lipids, proteins, and nucleic acids. And lipids is the same thing as fat. So when you hear the words carbohydrates, fats, and proteins, it sounds like the food you're eating, doesn't it? Well, you are what you eat. You need those molecules in your body so that you can grow and develop and maintain homeostasis. You're putting molecules in your body so that they can do that. We're not yet a living thing yet, because what I can do with my molecules is I can form these little tiny structures called organelles. And organelles are like small little organs for your cell. And those organelles are going to make up the cell. And that's the next level of organization. And that's the level of organization where we define as you're a living thing. So atoms and molecules, organelles, not living yet. We're building up in complexity. We're creating different patterns that we can examine in systems, but we're not a living thing yet until we get to number 3, and that is a cell. So I can be a living single-celled organism. I can be a multicellular organism. I can just be a bunch of cells that kind of live in a little colony. But as long as I have a cell, I'm a living thing. Now, as we get like bigger and we start thinking about, well, a human being, I have to organize myself up there and get a little bit more complex. Tissues are a bunch of similar cells performing a similar function, and so they allow them to do a higher degree of work that the individual cell couldn't do by itself. Now, if I take a bunch of tissues and of different kinds and put them together and they start doing a similar function, that gives me an organ, like your heart or your stomach or your spleen or your eyeball. Those are organs and they're doing a specific job in your body, like maybe pumping your blood around, but they're made out of different types of tissue, which are all working collectively to do their function as a tissue. If I take all the organs that are doing the same type of job, then I get an organ system. So if I have like your esophagus, your stomach, your small and large intestine, I now have your digestive system. And that's allowing those molecules you're eating, your food, carbohydrates, lipids, and proteins, to come into your body and be broken down to the molecular level so that your body can use them. If I take all of your organ systems together, now I got you. I have a multicellular organism, complex, awesome in its own nature, doing all sorts of neat things. And I have another level of complexity, probably one of the more complex levels. And I've been able to just kind of build up on that. This is as far as we go in biology, and we actually spend most of our first term at looking at these different levels and complexity. If you want to go bigger, then I recommend you take environmental ecology, where we start taking organisms and looking at them in terms of populations. We look at how those different populations interact in terms of communities. And if I take my communities in a given area and look at the non-living things like weather, temperature, oxygen, the dirt, I get an ecosystem. And the most important level organization is a biosphere, because that's all your ecosystems put together. That's planet Earth. And when you start understanding these levels of organization in the construct, you'll see why we really, really, really need to take care of our planet. So, if I need to eat, I need nutrients to maintain my homeostasis, where and all does that come from? Well, we eat things. That makes us a consumer. And we can consume plants and we consume animal-based products. Plants are considered producers because they can actually capture the energy from the sun and make it available to us. And our energy ultimately then comes from the sun. So all energy on the planet, all energy that all life needs on the planet comes from the sun. We just have to know the path, the way that it's going to get to us. So it starts with the sun. It is collected by producers, plants, which turn it into chemical compounds that we eat. We eat potatoes, we eat strawberries, we eat bananas, or we eat the animals that eat the potatoes, the strawberries, and bananas, and we take that food into our body and break them down into molecules that we need to do all sorts of cellular and chemical activities so that, again, we can maintain homeostasis. Those molecules get cycled. They don't get created or destroyed. They just kind of get moved around and around in systems called biogeochemical cycles, which we're going to look at in the next unit. But energy has a linear path. It goes through the system, and when it's gone out of the system, it's gone. It doesn't get recreated. Energy's used, it's one and done. So energy flows through our systems in one direction. It does not cycle back through organisms. So when we're out of energy, we're out of energy. We just have to get more, which is why we have to keep eating. One other characteristic of life is to help us maintain homeostasis is be able to respond to change, the stimulus all around you. You have this whole organ system, your nervous system, developed to help you respond to stimulus so that you can stay alive. And it happens in two places, external and internal. Internal, we probably really don't think about that when you sat down and ate to breakfast and you put food in your body, it was because your body was stimulating you, hey, you need to get those molecules inside. Now, once we get that food inside of you, those proteins, carbohydrates, lipids, and nucleic acids, they've got to be digested. So now you've triggered another stimulus for your body to break down those foods into molecules. So your stomach is now secreting acid and enzymes, and it's churning and mixing up your food so we can make this chimy stuff, this liquefied food, so it can pass into your small intestine and get digested there. External environment would be more of your flight or fight response to keep you alive. So if there's like a big scary shark and it's swimming up to you, your body's going to release a bunch of adrenaline into you so that you can deal with the big scary shark, that you can either stand your ground and have the strength to fight it or run away super, super fast. So that's the response to stimulus. But all of that is to keep you to stay alive, because if you're not alive, it's a sad thing. It all goes back to homeostasis, keeping that same state. I need to maintain a constant internal temperature. I need to make sure that my whole organism is safe and can respond or escape danger from the outside. So homeostasis, same state. A good example is your body temperature. You need to stay around 98.6 degrees. So if you get too hot, what do? You start taking off layers, you're sweating, you try to find someplace cool to get to so that you can bring your body temperature down. If you get too cold, you start putting on layers, you try to get inside where it's warm, and you start shivering because your muscles will generate heat for you to warm you up. If you go one way too hot, one way too cold, you die. Your body can't maintain yourself at those temperatures and you pass away. So let's make sure we can maintain homeostasis and not let that happen. Metabolism is the means to help you maintain homeostasis. When I was talking about you taking in all that food, carbohydrates, lipids, proteins, and nucleic acids, it's so that you can run metabolism. Metabolism is like your engine, and the foods you bring in let you fill up your tank so that you can keep your engine running. And it's basically what's the big word for saying, hey, let's make these cellular reactions happen. Let's make sure we're getting energy, we're keeping energy in our cells so that they can do their work and maintain homeostasis. Now we're going to look at one of the most important molecules in your life, DNA, right here. And you actually get the building blocks of DNA from the nucleic acids. So if you're ever wondering, like, what's nucleic acids? Nucleic acids make DNA, and that's why we consume them. So if your cells need to grow or divide, we have the resource to make new DNA. But DNA is what controls everything in your body. Every cell is told what to do or what it's going to be by the DNA inside its nucleus. So right here, DNA makes up these chromosomes. Chromosomes have genes on them. Genes are like the specific recipe for a specific function within your cell. So DNA, because it is like the control molecule, the recipe, the instruction book, the blueprint of life, guides all sorts of functions in your body. It guides your development. If you start off as a single-celled organism and we got to divide you through to get to trillions of cells to be a grown human adult, it's going to guide that. It's going to say, hey, you're going to be a heart cell, and you're going to be a liver cell, and you're going to be an eyeball cell, and you're going to be a skin cell, and you need to sit here and work with these cells so that I can form a heart, and you need to be over here with these cells so that we can form a brain, and I need to put you all together so that I can get a happy adult out of all this single-celled thing. It also controls the growth of the cell, the size, the volume, the number of cells. So to kind of help you figure out, well, what's the difference between growth and development? Growth is I'm increasing the number of cells. I'm getting bigger. I'm moving up in size. Development is maturing the cells or maturing the organism so that you go from like a toddler to a teenager to a full-on adult. There's a maturation process with development, where growth, I'm just adding cells. I'm making something bigger. It also controls reproduction. So when you hit development and you are a mature adult, we're making sure that those specialized structures are good to go and that we can pass on your DNA to your offspring, because DNA is the mechanism for inheritance. You are you because of the DNA you inherited from your parents. If you choose to have offspring, your offspring are going to be what they are because of the DNA you passed on to them. And so the neat thing about DNA is we can get little changes, and little changes is why we see little differences from person to person, population to population, and it gives us this broad range of diversity, which is awesome. But it also lets us do the next thing, and that's change through time. That's evolution. Evolution is based upon changes in your DNA. And it seems subtle at first when you look at an individual organism, but when you start looking at populations and you start studying populations over a long period of time, then you can see some dramatic changes in what something looked like or what something was 100 million years in difference. So populations evolve. We don't as a person evolve. Any single organism doesn't evolve, but a population of organisms will evolve over time. Take, for example, our friendly little dragonfly. Today we know it is something that's about palm-sized. We find it around marshy areas, but 100 million years ago, these things were 6 feet long with 6-foot wingspans. Why? Because they could survive like that. But over time, and changes in the environment, and as a consequence their DNA, they shrunk because the environment couldn't support anything larger than that size. It's pretty cool to think about, and again, this is a key thing we're going to start exploring and understanding better in term 2. So let's make sure we go back over our characteristics of life again. We know that living things are organized. We know they are made out of cells. I mean, that's the clincher. Is it made out of cells? Yes, no? Okay. But we have to respond to stimuli so that we can maintain homeostasis, and we use metabolism as our way to help maintain homeostasis. We grow, which means we get bigger. We develop, which means we mature, and we reproduce so that we can pass on our DNA, which controls everything. And that as a population, we are going to change through time. So let's make sure you know what you're doing. Bacteria, living, non-living, what do you think? Well, if you said living, you were right. Bacteria are living. They're single-celled organisms. And they're made out of a type of cell called prokaryotic cells, which we're going to get into a little later with in this unit. A sponge. That one's a little hard. You probably may have one sitting in your bathroom. You use it to wash off with, but did you know it's an animal? It was once an animal. It actually belongs to the kingdom animal. So yeah, it was a living thing. What about fire? Fire grows, it consumes things like metabolism, but the big clincher is, does it have cells? No. So no, fire is not a living thing. What about kittens? That one's kind of easy. Yeah, we know that kittens are living things. What about a virus? This one's not so easy. We need to think about all the properties of characteristics of living things and decide, is a virus a living thing? And I'm going to tell you no. A virus is not a living thing. And here's why. Viruses lack the characteristics, all the characteristics of living things. You have to have all of them, all or none. And although viruses look like cells, they have parts that might be considered cell-like, they are not living because they do not have, they're not made out of cells. They lack cytoplasm, which I know I have to have inside my cells. They don't have organelles, which cells have, so they can carry out functions. So because they don't have organelles, guess what? They don't carry out metabolism, and they don't have to maintain homeostasis. So that's like a really big deal. Homeostasis, it's not happening. And the other thing is, although they do reproduce, they don't reproduce outside of a host cell. They actually have DNA. It can be RNA or DNA, and they actually stick it inside the cell, and that information travels to the nucleus of the cell and gets in there, and that's how they get the cell to make more virus. And then they fill up, the cell will fill up with viruses, and then it will blow up. So if you ever get a virus and it's infecting your cells, now you know those viruses are blowing up your cells, and that's why you feel so miserable. So what can we do about it? Well, if some viruses are so deadly and they're so risky for humans that we create vaccines to protect us against them. And how that works is I have either a dead or an inactive virus that we put in the vaccine. We give you the shot, like your flu vaccine, and it stimulates an immune response in your body so that your body's going to say, hey, it's a virus, get it. And your body will learn that, oh, it's the specific virus, it's virus X. Well, now I have the immunity to fight against virus X. So if you're ever actually exposed to virus X in the real world, you won't ever get sick. Why? Because you have that immunity built up. So if that virus ever tried to re-enter your body, your immune system would just like pounce all over it and crush it to death, and it would never have a chance to make you seriously ill. So here they are, your characteristics of life. You got to have organization. You got to be made out of cells. You have to respond to stimulus. You have to maintain homeostasis. You have to have metabolism, which is you eating food. We're going to grow, you getting bigger. Development, you getting mature. And you're going to reproduce so you can pass on your DNA. And then as a population, we're going to change through time. There are some microscope basics that I want to hit upon to make sure that you understand how to use a microscope. We're going to go through all these parts in class to make sure you're comfortable using the microscope, but I want to make sure that you understand how to determine your magnification. And so to do that, you multiply the power of the ocular lens, which in this example is 10x, by the power of the objective, which here is 40, and that gives you the magnification. So 10 times 40 is 400. So I am making, what I'm looking at is going to be blown up 400 times than what it normally is. And it makes it so much easier to see these little things under the microscope. So it's a lot of fun to play with, and we're going to get a lot of opportunity in class to use the microscope. So when you have a chance, look at the different powers of objectives on our microscopes in class so that you can figure out how much it's being magnified. Okay, so what's the difference between resolution and magnification? Well, or what's the difference between the different magnifications on our microscope? Well, when you look through the eyepiece, that's your field of view. So what you see in that little circle is your field of view. And depending on what eyepiece objective you're looking at will depend on how magnified it is. So here's an example of looking through the microscope, field of view under low power. I can see a lot of field per se, but everything's still kind of small. If I go up to high power and I increase my magnification, I can see this cluster of cells from here over here. I can see them really big now, but I lost a lot of my field of view. So high power, I can see smaller things in a bigger, more detail, but I see less field of view. So I need to keep that in mind, so when I go to try to find something, I keep what I want to look at completely centered in the field of view. Because if I wanted to look at this point over here and I go to high power, all of a sudden it's clear off screen. I can't see it. And it's really hard to move the slide around on the microscope to relocate that. So it's better to start off on low power first to find what I want to look at and center it. So one thing to remember is always start on low power because it has a bigger field of view. And then adjust your microscope to get up to high power so you can see the details that you want. All right, we should be good. If you have any questions, don't be afraid to ask me in class. And again, use your resource, your textbook, to help you with the information provided. And you should be reading Chapter 1 anyway.